I'd like to introduce Clare Trumble. Clare is an associate genetic counsellor with experience across both public and private sectors. She specialises in familial cancer, clinical, prenatal, metabolic and reproductive genetics. Currently working as a reproductive genetic counsellor at Genea Fertility, she has a focus on helping individuals and couples navigate their genetic options and supporting them in understanding and adapting to their results. In this role, she discusses reproductive options, for couples with hereditary conditions, including IVF with pre-implantation genetic testing. A key part of this role is ensuring couples understand both the potential and the limitations of the testing. Now, finding the interesting facts about all our speakers, are you finding them interesting? So, Clare's claim to fame was when she was in primary school, she was in an aerobics team called TPAC. They wore those awful pink and black spotty leotards with spotless white trainers. Clare said that it was really hard work and she's never been able to do a push-up that well since. They made it to the national championships and they won gold. Please welcome Clare.
Hello everyone, good morning. Thanks for being up and bright and bushy-tailed to listen to genetics. First thing Sunday morning, daylight savings. I'm very impressed you're all here, appreciate it. Yes, I'm Clare, I'm a genetic counsellor. I'm going to talk today about genetics and specifically the genetic testing that's available at different parts of pregnancy, but particular pre-pregnancy. So the aims of today are to understand how genes can cause OI, talk about some of the reproductive options, and then in particular discuss this type of genetic testing. So it's called pre-implantation genetic testing. So just to give a bit of an idea about where I come from, what the genetics landscape looks at the fertility company that I work with, Genea. So we have the genetic counsellors and then we have our genetics doctors who are the geneticists. We also work with the fertility doctors who are the fertility specialists. Then there are the nurses, the counsellors, the donor coordinators for people that are using donor or egg sperm, and then we liaise with the outside doctors and GPs as well. The kinds of things that we're talking about with our patients are carrier screening, so that's a type of genetic testing that's done on parents planning to have a child. We do genetic testing of embryos, and then we have egg and sperm services. So that's kind of the context of where I'm coming from. So we get people that come through who have genetic conditions, serious genetic conditions, that they're interested in kind of exploring what the options are for them. So if I had a dollar for every time someone asked me what a genetic counsellor does, I'd be a very rich lady. The kinds of things that we talk about are helping patients and families to understand and their genetics and what their options are. We provide support for patients and families that have a genetic condition at different parts of the process where it can be challenging. And we help patients and families to come to a decision that's right for them about testing, about what options they're going to do following a testing result. So it's all about choice. It's not about saying this is the way that you have to use this genetic information. It's just about making people aware of what their options are and helping them to kind of think about what might be right for them. So this is where the biology lesson really kicks in, so buckle up. And I apologise for the people that this is very familiar to, so you can zone out from now for the next 5-10 minutes, but I think it's helpful to understand how the genetics is actually causing OI to understand how this type of testing works. So our bodies are made up of millions of cells. Inside all of our cells, we have a complete set of our chromosomes. So our chromosomes are made up of DNA. DNA, which you can see on the right. Let's see if this works. It's a code, and it's a code that our body reads, kind of like the set of instructions for how everything in our bodies works and functions and grows. So the strand of DNA code is wound up into the chromosome. The chromosomes are what float around inside the cells. And this is what one full set of chromosomes looks like. So you can see that all of the chromosomes come in pairs. So for example, chromosome 1, we've got 2 copies all the way up to chromosome 22. So in general, chromosomes 1 to 22, males and females all will have 2 copies. And then we have our sex chromosomes. So males have an X and a Y chromosome, and then females have 2 X chromosomes. And this becomes helpful to understand when we're talking about what type of OI you have and how it's inherited, because some of the genes are located on the X chromosomes, whereas some are on the autosomes, or the non-sex chromosomes, basically. So that can affect males and females differently, because males have 1 copy of all of the genes on the X chromosome, whereas females have 2 copies of all of the genes on the X chromosome. So a gene, it's a section of DNA. So if we think about the DNA as the full set of instructions for how everything works, a gene is 1 instruction. And it's an instruction, or some people call it a recipe, for a protein to be made. The protein is it has some sort of role in our body, so it might be floating around in our blood. In our eye, it's usually got something to do with how the bones are formed. So when we get a change in the DNA code, so if I just jump back here, so you can imagine that it's a change in that spelling on the right-hand side. When that change happens, it can cause the protein to be not properly made, it might be absent, and it depends on what type of genetic change we're seeing. So how does OI occur? So it's most often caused by a change in COL-A1 or COL-A2, but as Professor Lentz was talking about yesterday, it can get very complicated, and there are so many different genes which can cause OI, and it can be that it's inherited in different ways, which we'll go through. So just to give, I'll use collagen type 1 as an example to show how this works, because it's the most common. So on the left, we've got the 2 genes that are the instructions to make 2 parts of the protein, which is collagen type 1. So COL1A2 is the instruction for the alpha-2 chain. COL1A1 is the instruction for the alpha-1 chain. These are all wound together into the triple helix, which is collagen type 1, which goes on to form the bones, the skin and other tissues. So when there's a mistake in one of those genes, then it can cause faulty or missing protein. So there's a lot going on here, but I just wanted to, on the right, show what the different types of genetic changes can be, because as I'm sure you've seen, the same gene can cause multiple different types of OI, and so to understand how that can occur is the difference between the different types of genetic variants. So this one is showing that some of the code has been deleted, some of the code in this option has been added. Here there's been a bit that's been taken out and a bit that's been added. And there are a number of different other ways in which the DNA code can change, and depending on how disruptive that change is, will affect how much that protein product is changed. So if it's only a very mild change to the DNA code, the change to the protein might only be mild as well, which then in turn can cause a mild presentation. If it's the type of change which is, it's stopping the DNA code and the rest of the gene isn't there anymore, that can cause a much more severe presentation. So on the right we've got, and I simply could not fit all of the different subtypes in, So I've just taken a section out, and it just shows that there's the same gene which can cause different types of OI, and then it goes the other way around. So the different types of OI can be caused by the same gene. I think I said that the same way around, but it can go both ways. So there are different ways, of course, in which OI can be passed down. So the first way that we'll go through, which is the most common, is autosomal dominant. So this is where, and I should highlight that we've all got 2 copies of all of the genes. So I showed that we had 2 copies of all of the chromosomes. The genes are located on the chromosomes, so we've also got 2 copies of all of the genes. We get 1 from each parent, and then when we have a child, we pass on 1 copy, and it's completely random which copy gets passed on. The child will get the other copy from the other parent, so that they also end up with 2. So with autosomal dominant inheritance, you just need a variant in 1 copy of the gene in order to have the condition. So that's what's shown on the right here. So in this case, it's the dad that has OI. He's got a working copy of this gene. It could be COL1A1, for example. And then he's got 1 faulty gene, which is shown by the blue line. And then the mother she doesn't have OI, so she's got 2 orange, 2 working genes. So each time they have a child together, the mum is always going to pass on a working gene, and for the dad, it's 50-50, that he passes on the typical gene or the gene that has the OI variant. So you can see that half the time the child will have OI, and the other half the time the child will not have OI. And in this case, it doesn't affect male and female, it's the same. So it's 50-50 regardless of sex. From that generation, any child that has OI would also have a 50% chance of passing on, and any child that does not have OI is not going to pass that on to their child. It's not going to skip over them. They don't have the faulty gene, so they can't pass it on. That said, it can be variable. So this is why we see that in some cases there might be a child who's diagnosed and the parent has genetic testing, and it's only at that time that they get a diagnosis, because they might have a more mild form of the condition. So even in the same family, the same genetic change can have a different presentation. What we also see sometimes, and it varies between different genetic conditions, but we do see it happening in OI, is that we can get a completely new change that's happening for the first time in the individual. So it's not been inherited from either parent, it's a new genetic variant that's popping up. So we often see this sort of presentation where there's one child who has OI and neither parent has it. And in this case, we wouldn't expect any other siblings to have it. So if there is a truly de novo change, then the risk of having another child with OI it's not increased to anything above the population risk. Where this can get a little bit complicated and always has that element of uncertainty when we see that there's 2 unaffected parents and 1 child who has OI is the possibility of this, which you might have heard of before. So it's called gonadal mosaicism. So this is where there is a change that exists, so a genetic change that is not in, it's not in the blood cells, it's not in the hair cells, the skin cells, it's only in the egg cells for a female or the sperm cells for a male. That's pretty tricky to pick up, so it's not something that we can easily test for. They usually won't have OI, so it won't be evident. It'll only be apparent if they pass that gene on and they have a child who has OI. So it can be a similar family structure to what I talked about with the new change, where you've got 2 unaffected parents and a child with OI. But the difference with this one is that because the change is still in the egg cells or the sperm cells, it can be passed on a second time. So it means that we might get a second child who has OI with 2 unaffected parents, and if you were to do genetic testing on those parents, you wouldn't pick up that they had the genetic variant. I think that the incidence, it's tricky to estimate because it's hard to test people for gonadal mosaicism, but it's roughly around 10%, but it was variable from what I could see. So less common for OI, but is still a way that it's passed down, is autosomal recessive inheritance. So this is where, of the 2 copies of a gene, if you had one copy that had the genetic variant in it, you would not have OI. You most likely wouldn't be aware that you're a carrier. It would only be if 2 carriers of OI had a child and they both passed on their genetic variant, so that the child ended up with 2 genetic variants, that child would then have OI. So in this case, we can do genetic testing of the parents to see that they carry those 2 variants, but they wouldn't themselves have OI. So the chance of 2 carrier parents having a child with OI would be 1 in 4. And again, that's regardless of sex. So there's a type of genetic testing that parents can access. It's called reproductive carrier screening, where there can be just a few conditions that are looked at, or hundreds of conditions that are looked at that are associated with recessive conditions. And sometimes OI is included there so that people know ahead of time whether there's a chance that they're going to have a child with OI. So good to be aware of. For example, if you have a sibling who's interested in having a child and they don't know whether or not they carry the OI gene, but it's just good to proceed with caution, because this isn't a test that's looking at all of the OI genes and taking a good look. It might look at only some. So if you do have a family member who's interested in being tested for the OI gene that you carry, just make sure that they have a chat with a genetic specialist first to make sure they're getting the right test and it's not going to be looking at a completely different gene. So bear with me, we're almost there. There's lots of different ways in which it can be passed down. So X-linked inheritance, there's a few genes that can cause X-linked inheritance with OI. So as I mentioned earlier, females having 2 copies of the X-linked gene, of the X-linked chromosomes will have 2 copies of all the X-linked genes. This means that they can be a carrier of an X-linked condition, and in some cases they might be unaffected, in other cases they might be more mildly affected than a male who has a faulty gene in their only X-linked gene. So if you have a mother who is a carrier of an X-linked condition, the chance of having a son who has the condition is 1 in 4, and then a 1 in 4 chance of having a daughter who's a carrier. So for some of these genes, a female who is a carrier, she'll have osteoporosis, but might not be as severely affected as a male. That can be pretty variable because of something called skewed X inactivation, which is basically where if you are a female and you've got 2 X chromosomes, usually only one is sort of turned on at a time, or it might be turned on for good. So if you have the one that's turned on that has the fault in it, then you might have a more severe presentation. If you have the other one turned on that is a working copy of that gene, then you might be more mildly affected. Okay, we're over with the inheritance part you got through. So there are many different reasons why someone might do genetic testing. It can be to do with understanding what's going on and why you've got a collection of symptoms and sort of bringing that all together to get an answer of why. It can be to tap into wonderful support groups like this. It can be to help with management, and as we spoke about yesterday, with precision medicine, making sure that you're getting the right testing for the gene that you carry. And then what I'm particularly interested in, and what we'll go into now, is the reproductive options, and understanding what's the inheritance risk and what options are available to me. So there are a number of different options, and as I As I mentioned at the top, it's not up to us to say which one is the right option for you. It's just about making sure that you're aware of all the different options and what are the benefits, what are the limitations of each. So there is the option to do testing pre-pregnancy. Some people will do testing during pregnancy. So that's typically via, it's called CVS or amniocentesis. I won't go into it in too much detail, but that's testing from about 11 to 15 weeks pregnancy to see whether the baby, so to do genetic testing on the baby. And then there's the option to do testing of newborns or to not do testing at all. So pre-implantation genetic testing. So pre-implantation, it means it's before an embryo has been implanted in the uterus. So it's going through the IVF process to develop an embryo and doing testing before the embryo is being transferred. There are many different types. The type that we're interested in is PGT-M. M stands for monogenic, which means a single gene. So in this case, it's looking at the gene for OI. So it's a genetic test that can be performed in embryos created via IVF, where an individual or couple are known to have an increased risk of a specific single gene disorder. It's very important that we already know what the gene is so that we know what to look for in the embryo, and it allows you to significantly reduce the risk of having a child affected with a single gene disorder that has been identified in your family, so such as OI. So when we're starting the process of PGT, we need to get that genetic testing report. Ideally, we get a clinic letter. So if you've been seen by a genetic service, getting a copy of what they've tested and what the interpretation is really key. And then we do need to get blood spot samples to be able to test the DNA of, so that will be for both partners. And then ideally, it's the parents of the affected individual, or it might be the couple's child or another affected relative. So testing multiple generations is going to increase the accuracy of the test. So it starts off about 90% accurate if you don't have that multiple generations of DNA samples. If you've been able to get the parents to test as well, then it's a 99% accurate test, which is really helpful for couples when they're making such an important decision about which embryo to transfer. Of course, it can be tricky sometimes if parents are, if they've passed away, if they've lost contact, if the child doesn't want to include the parents in the process. If they are overseas, then that's actually okay. We mail the blood spot kits overseas. They'll prick their finger and post it back. So yeah, just something to be aware of. So it is a bit of a timeline. There's a few steps involved. It'll start with seeing the fertility specialist, which is a doctor that specialises in fertility. Then we start the intake with the lab. So that's getting information about what the condition is, getting the report, figuring out who might be available to provide a DNA sample. You'll meet with a geneticist doctor. You'll have other consults. And then if you decide that you would like to proceed with the testing, then that's where you can make that decision after you've had those discussions. Once you've decided that you would like to go ahead, then the workup starts, and that's where the scientists are building the genetic test that is specific to your genetic change and your sort of who's provided samples. Once the workup is complete, then you can start an IVF cycle. So you might have heard women who are doing an egg collection or going through IVF, they do injections, so that's when those start. That's to stimulate follicle growth or egg growth. They collect the eggs, they fertilise the egg, so put the sperm into the egg, and then the embryo starts to grow. So we've got these really cool sort of incubators where the embryo sits at Genea, and they've got a camera so you can actually see the divisions happening. So it'll go 1 to 2 to 4 to 8. And then after 5 days, you've got a little embryo it's called a blastocyst at that stage, and it's about 100 cells. They take a biopsy or a small sample from the outer cells so that they don't disrupt the inner part of the embryo, and then they take the DNA out of those cells, and that's what they're testing. That part of the process takes a couple of weeks, and the kinds of results that we get back are either that the embryo is unaffected, so it's inherited to working copies of the gene. It could be that the embryo has the condition, and then in some cases, because they're working with such tiny, tiny bits of DNA, sometimes it's not possible to do the test. And so in that case, it might be that we have to take another sample, or it might be that the couple just won't know what the result is of that embryo. So then once they have an idea about what the results are for each embryo, the patient will decide which one they want to transfer, and that one gets transferred back into the uterus. Easy as that. There are a couple of different ways in which the genetic testing happens. So sometimes the lab is just looking to see if the genetic variant is there. So they look at the DNA, is the mutation there, is it not there? That's the test that's about 90% accurate because, again, because they're working with such small bits of DNA, it's possible that they look at the sample, they say the variant's not there, but it's actually that it's just missing from that small sample because it's been deleted out for some reason. So to make it more accurate, there is this process called linkage, and I won't test you on this one because it can be a bit complicated, but it's just to understand that this is why we need to get the multiple generations of DNA, because what we're trying to do is understand what does the chromosome look like where the genetic variant is. In this case, it's the red chromosome. So we can kind of track it through the family. So we know what it looks like in the parents of the couple, we know what it looks like in the affected couple, and then when we see an embryo that has that particular chromosome, we can be pretty sure that it's going to have the genetic variant, even if we can't see the genetic variant. If we're seeing that the embryo does not have that chromosome, we can be pretty well sure, 99% sure, that the embryo is unaffected and it's a good embryo to transfer. So there are a few different things to consider with IVF, and I haven't put here that, there's things to consider personally, whether it's something that you want to test for, it's something that you're wanting to know what the results are for your embryos to make that decision at that point. It does require the process of IVF, so rather than kind of the spontaneity of conceiving naturally, it does medicalise the process of becoming pregnant. It requires involving other family members. There are additional costs and time frame as I've stepped through. For the costs, there have been some Medicare rebates that have come in the last few years, which is really good. There's still a way to go, so there's usually still a gap if you're going privately. I actually don't know what is available in Adelaide. In Melbourne and Sydney, they've got public services available where you can do the whole process and it's completely funded, which is awesome. But again, they're pretty busy at the moment, so there's still a way to go there. Taking the biopsy, doing any test to an embryo, there is a small risk that it's not going to survive the process, but that risk is pretty small. Given there is not that 100% accuracy, we always talk about the option of doing testing during pregnancy in addition to doing testing pre-pregnancy, just to get that extra level of understanding about what's going on for that baby, whether or not they have the condition. So that's a personal decision. Some people will say will be comfortable with the level of accuracy and comfortable with that small chance of it being a discordant result, whereas other people would like to do every test that's available. It is a numbers game. So what I mean by that is that there is an element of luck involved. So whilst we can test what the embryo carries, we can't create an embryo that has a particular genetic result. So it relies on having an embryo there that hasn't inherited the condition, and that also doesn't have any other chromosome changes that might. So if I should say as well, when we test the embryo for the genetic condition, we're also looking for other chromosome changes, and it's particularly common as women get older that you'll see a chromosome change that's going to severely reduce the chance that the embryo will be a successful ongoing pregnancy. So it could be that you get 5 embryos, 4 are available to test, and then 2 might have the genetic condition, 1 might have another chromosome change, and then you've got 1 embryo left to transfer. So it's a good outcome, you've got an embryo to transfer, but sometimes people will have to go through multiple IVF cycles in order to get an embryo to transfer. It can be emotionally challenging, particularly if people go through quite a lengthy process to get a result back that shows that they don't have an unaffected embryo to transfer. So having the supports available from the get-go, we've got counselling support that's available for people going through IVF and PGT whenever they need it. So that's really important to not kind of diminish that part of it. The benefits are somewhat more self-evident and can be pretty powerful, particularly for people that have themselves or in their family been seriously affected by a genetic condition and want to avoid passing it on. It can offer a sense of control and reassurance where otherwise there might not have been. And then there's also that added benefit of checking for chromosomal changes. So that really increases the chance of a successful ongoing pregnancy, is the benefit of that one. So if you are, or you've got someone in your family who's interested in looking into this further, first step would be to talk with your GP. Next step would be that they would refer you on to a fertility specialist. You would need those documents, so if you don't have a copy of your genetic testing report or letter, then reach out to your genetic service and they should be able to hand it over to you. And then once you've met with a fertility specialist, they'll refer to the genetics team and we'll take it from there. In South Australia, we've got a number of fertility specialists, and we're located all over Australia in most states, so you can find out a bit more on the website. That's everything I had to go through, but I think Katie might, where has she gone? There she is, might have a chat about what it's like to be on the other side of it, going through the process. So I'll hand over to you.
Hi everyone. I know that I'm only going to speak briefly because I think it's really important for people to be able to ask some questions for Claire, if that's okay, if you want to hang out up here. I've met a lot of you in the past and also at this conference, so I'll keep it fairly brief. My husband and I went through the journey of IVF a few times, and I'm really grateful for Claire to mention not just the whole clinical information about the options available, but also just sort of say that it's quite emotionally taxing, because I think that a lot of people are very practical about, child rearing, etc., but you sort of don't think about the emotional toll that all this process actually, because we live with OI, some of us don't have OI, but we have family members with it. And I think that it's just important to know that what Claire is mentioning, it's all about choice. And this, is just so amazing that we have all these different choices now, but it is very overwhelming. So take your time with wherever you're at with it. And I think that, there's some great counselling services through a lot of fertility specialties now, which is really important, and also just external. So, I always knew that I wanted to go through testing just for myself personally. My father has OI type 1, COL1A1, and my brother also has it, and we've both had our own interesting journeys through pain and suffering, and obviously watching my dad over the years as well have his issues. And even before I met Andrew, I knew that I'd want to have a child that doesn't have OI if that was an option. It was obviously a very long time ago, and the options have changed constantly since that decision was made. We went through our first cycle, and I was very in denial about the numbers because we've got a 50% chance of passing it on. And I thought, well, that means that if I get 10 eggs collected, then, I'll get 5 for testing. And then, that's really amazing. And my husband's, who's very numbers and statistically appropriate, was like, Toots, that's not really how it works. Remember, there's like all the testing, they have to be appropriate for testing, and they have to be, and I was like, oh yeah, that's right, yeah, okay, yeah. So we had a really interesting journey with our first cycle. We had 3 embryos for testing. 2 of them were affected with the OI and 1 was unaffected, so not chromosomally affected with anything else because I was a bit older at the time as well. And that turned out to be Cooper, who's now 6, and that was pretty amazing. I felt like the luck of the draw was really good. Second cycle, after deciding that we weren't just going to have 1 child because, you know, life was not as difficult as I thought, being an OI mum, but it was still came with its challenges. Second cycle, we had 5 embryos for testing, so I thought, okay, yes, this is going to be perfect, because 5, we only had 3 in the beginning. But out of those 5, 2 were affected with OI, and then the remaining 3 were chromosomally abnormal. So that was really hard. And I think that's where the emotional support and counselling and things like that were just like, you know, I'm so glad that we had that information, but it was really difficult because I thought, wow, okay, well, what are we going to do now? So, you know, we took a step back and had some time to ourselves for a while, and I thought, and I talked to Andy, and he was very much just leaving it up to me to decide where we wanted to go with it. And I said, look, I think that we just give it one more go. You know, one more go, and regardless of the outcome, no more after that. Because it is a lot, you know, very clinical, like Claire mentioned, you know, it's not like, oh, so you're pregnant, and it's... Andrew wasn't even there when we had our embryo transferred because he had to work one day. So it's kind of strange, but it's also amazing. So our third cycle, we ended up with 4 embryos for testing. 2 were affected with OI, and then we had 2 unaffected embryos. And that second, or the third and final cycle, we got our son Angus, who's nearly 3. So it is a lot, but I'm just grateful that technology gives us so many different options. Whether you do the pre-pregnancy testing, whether you do the testing in pregnancy, whether you don't test at all, it doesn't work for everybody. And I think that that's really important to note, that you don't have to do this, but it was a really great decision for us, and I'm really grateful that we could have the journey. And yes, being a mum with OI is really hard. But you also teach your children that don't have OI how to be more careful. And both of my children, if I say ow, regardless of where they are in the house, you okay, mummy? You know, even Angus, he comes running and he'll give you a big cuddle, you know, because that's how... And I think that that's lovely. So we're teaching him to be careful with mum, because they are going to be gigantic humans. My husband, as some of you might remember, he's 6'4", so I basically work to feed my 2 humans because they eat so much food. But I'm happy to have a chat to anyone at morning tea if you want some more information about it. You know, I'm a nurse as well, so I'm very happy to be, you know, clinically open about the ins and outs of it, because it's a lot. And I know we've got a few people that are a bit younger and just want to know the options out there. So, and we also have Evie and Matthew are up the back. And I think it's really important to know that Matthew, for the gents in our room, he's also quite happy to have a bit of a one-on-one chat, because I think it's really important. It's not just affecting the women, it's affecting the men as well. So, yeah. So I'll leave it open for some Q&A with Claire, if you guys are happy with that.
Yeah, thank you. Any questions?
Yeah, I've got one.
Yeah, go for it.
You talk about when you're testing and having the parents and the grandparents, the parents. Does it affect the chances if you get more and more generations?
No, they only need 2 generations. Sure, of course, yeah. So the question was, when I was talking about needing to get the parent sample, does it help if you get multiple generations? And it's just the 2 that you need to get that 99% accuracy. If you can only get 1 generation, like if it's only the couple that's available to test, initially it'll be a 90% accuracy, and then after 1 round, sometimes the scientists can basically use the results of the embryo testing in order to boost the accuracy up to 99%. But yeah, it'll be maximum 2 generations that you'll need. So if grandparents have passed away, then that's fine.
Did you say if you get the siblings as well, that increases?
Yeah, so if you get the siblings, does that help? So it needs to be one other affected family member. It helps the lab if it's sort of multiple generations, but they can do it if it's a sibling as well.
Thank you. So if you were to do genetic testing with embryos, could you also, if you weren't trying to void OI, are you able to check the severity of the embryos?
So it'll be the same gene variant that's been passed down from the parent.
Okay, so that's always.
Yeah, and so mostly yes, you'll be able to know what the change is. It can, I guess, vary sometimes between males and females and between family members in general, just because of that variability. But you can most of the time get a pretty good idea.
And also I had another question. If 2 people with OI are trying to have a child and they have 2 different types, are like one parent more dominant, or is it just like a draw?
They'll be passed down separately, so you can almost look at it as two separate conditions that are being passed down in terms of the risk of passing down one and the risk of passing down the other, and then whether they combine to make a more severe presentation will depend on whether they are operating in the same pathway, in which case there wouldn't be an additive effect. So having two variants might have the same effect as just having one. And then again, it could be that having the 2 different variants, if they're operating in different pathways, it might be that they combine to make a more severe presentation. But it'll depend on what the variants are, and so it'll be, yes, very, very case by case.
I'll try to make a question. Hi, so we've got 2 boys. One of them's got spontaneous mutation, so we have never been tested. Would you advise for his sibling in the future to be tested in case he's carrying the gene, because I might be a carrier as well?
There's so many different possibilities with how it can with OI. So presuming it's a dominant type gene. So if neither of you have any symptoms, then the possibilities are that it's de novo or that there's gonadal mosaicism, and then if there's gonadal mosaicism, there is a chance that it's passed on to another child. Sometimes, so there are different approaches and it can vary depending on the genetic service. Sometimes the approach is if there is a chance that the child has the condition, then test kind of preemptively. Otherwise, it might be only if they have symptoms and they've had a few fractures, test at that point. For the sibling, yeah, only if it seems possible that the sibling might have OI, then test to confirm it. It can depend a bit on the genetic service and your preferences as well. So if it's something that you're interested in, have a chat to them about it and say, is there benefit in getting the sibling tested? Sometimes the first step is to get a clinical examination before going to genetic testing. So in the sibling, it might be that they go to see a specialist who will do an examination and then from there say whether or not they seem to fit with the type of OI that's in the family.
There's definitely no symptoms.
Yeah, yeah.
I was just wondering if he can be a carrier because I could be.
Yeah, it depends on what type, whether it's dominant or X-linked or what type it is. If it's dominant, then it sounds most likely that he doesn't have it. But yeah, have a chat with the genetic service and they can kind of tailor the advice to the specific variant.
I'm not a geneticist, but I just wanted to add a bit of clarity, I think, to the previous question. My understanding of the current literature around OI is that Prof yesterday made it quite clear that the type of OI that you have does not necessarily suggest the severity of the OI. So we know that the gene mutation specifically doesn't necessarily map to the severity as presented. The phenotype and genotype don't always match. So just knowing which gene is affected doesn't necessarily give you a sense of how the child might come out. We can look at databases of gene and mutations, and we can compare that to the severity of the OI as presented, and we can see mismatch. In fact, we even know that from my wife. That's the first thing. And the second thing is that children today born with OI are typically treated with bisphosphonates very early in life. And Prof has started to use this term bisphosphonate modified OI because the presentation of their symptoms is vastly altered. And so you can't just look at what your symptoms look like and project what the children's outcomes might look like, because even today, young adults who are potentially wanting to have kids will have very modified views of what OI would look like, and without that modification, their children would be substantially worse. So just looking at the genes is not necessarily sufficient to give enough information for what the outcomes might look like.
Yeah, absolutely. Having a good understanding, and I'm sure that people in this room would have a good understanding about what are the treatment options that are available, and then that could definitely affect the decision about whether to do PGT or whether to proceed, and know that there are good options available.
I've got a question, or a comment and a question. I don't know if it's common practice or not, but this is for families with, teenagers or even younger children. When we had our genetic testing, they also did the chromosome full testing. So at that stage, when our daughter was maybe 14, we found out she has recessive genes as well. So I encourage families to ask for full testing while you've got it available for free through the public health system or whoever you're seeing, because you can find out a lot of information early and be prepared more. And the other thing is more about, I was going to ask, do you suggest in Katie's case where she had 2 unaffected genes, people would freeze their eggs?
2 unaffected embryos?
Yep, sorry, yep, yeah. Yeah. So anything excess that's healthy, you would freeze. Yeah.
Yeah, absolutely. The usual process is to collect all the embryos and freeze them and then keep them there whilst testing happens, and then any embryos that you want to hold on to, if you're possibly considering them transferring at some point, they can be kept frozen for a number of years, and they'll survive that process pretty well.
Have you observed any significant difference between women who have OI and are still on biphosphonates or have had biphosphonates while trying to get pregnant, and women who have never had biphosphonates at all with their OI trying to get pregnant? Have you noticed anything at all with that?
I probably don't have the kind of the data set to be able to notice any differences, so I'll leave that one to the treating doctors.
I just had a question about the de novo and the gonadic mosaic spontaneous mutation. Is there a way to determine which one it is, where it's completely spontaneous versus the gonadal one? You mentioned it was hard to do pre-testing for gonadal.
Yeah, it is tricky. So in theory, if you could, do genetic testing of the sperm of the male, then that might be one way of being able to figure it out. But usually the only way to know is if they happen to have 2 children who have exactly the same genetic variant, and then you can assume that it's come from the eggs or the sperm. But otherwise, it can be tricky to test for in just sort of a clinical setting compared to a research setting.
Are there any further questions?
Hi, our daughter is quite young and has had quite a lot of treatment to begin with, which is fantastic to give her a good start. But, moving on when she gets older, we're trying to get as much advice to her to do this sort of genetic testing. And would it be best to discuss with her when she's a little bit older to maybe start collecting eggs and freezing even prior to thinking of children so that she has much healthier eggs to begin with? Because obviously age can affect the eggs as well.
Yeah, I think starting earlier definitely helps because as females, we're born with all the eggs that we're going to get, and as they sit there, they're more prone to developing chromosome changes. So the earlier that you're either collecting and freezing eggs or collecting eggs and then creating embryos to freeze, there is a lower chance of chromosomal changes. But I think, that's not to say as when she's 20 that should be something that is happening. It's up until about 30 is probably the chromosome change risk is relatively the same. So maybe from around that age or a little bit earlier, start having those conversations, and then depending on where she is, can figure out whether egg freezing is right or whether to wait and then create embryos and do the testing at that time. So I don't know if that's very helpful, but it can be a little bit case by case.
Look, I think we might call the question time to a halt there. And if you've got any further questions, maybe have a chat with Claire at morning tea time.